is my protein structure. My hand is my protein structure. Okay. Now I am, if you might be aware, people from biotech background or people from microbiology background, if you might be aware of this fact that protein, okay, protein folds and takes up a secondary structure. Okay. So when this protein, if you see this protein, like I'm using my hand to basically represent the protein. So if you see this protein, it is folding, okay, and then it takes up a secondary structure, okay, or maybe a tertiary structure or anything. It takes up a complex structure, okay. So it forms some pockets inside, okay. So protein is bound to have some pockets inside it. Now these proteins can serve as receptors also, okay. By receptors I mean some entities, or we can say some things that exist in our body to which endogenous ligands and exogenous ligands can bind to, okay. Let's just mention this here. So if we have a protein like this, who is coiled, okay, which is coiled here, okay. So I will have two things binding to it, okay. So first will be endogenous ligands and exogenous ligands, okay. So I can have these two things binding to it. Next, what I can do is now, whenever these ligands bind to a protein, okay, where will they bind? They will bind to some pockets which are present inside the protein, okay. So they will traverse a pathway, or they will take a passage, okay. They will take a passage inside the protein and they will try to sit within the protein, okay. So they will try to sit within the pocket that is there in the protein. Once they sit, okay, they will try to make some interactions. Okay, now what do I mean by these interactions? Now you might be thinking that interactions, okay, you have a, are you not able to see it properly? Full screen. Uh, is there any issue or something like that? Okay. You can pin, sir, you can pin it so that you can see it properly. Yes, sir. Yes, sir. If you have any other problem, you can please write it in the chat box. Okay. Yeah. So where I was is when this ligand comes inside the protein pocket, okay, it tries to do some interactions. Now, what do I mean by interactions? Protein is made up of many residues. Which residues? Amino acid residues. So these amino acid residues, which I will represent as A, okay, amino acid residues. So my ligand, suppose if this is my ligand, let's say a simple methyl or a simple methane molecule, okay, CH4, and let's say that this is a pocket inside the protein, okay. So now, if you can see it clearly, there are some residues which are going to interact with it, okay. Residues, which residues? The amino acid residues, because protein is made up of amino acid chains, right? Amino acid coming together, making a peptide chains coming together, making an entire protein, right? That is what we know. So when this methane molecules comes inside the protein pocket, what it does is there are some hydrogen bond acceptors, okay, and methane molecule might be having some HBA and HBD, that is hydrogen bond donors. Okay, so let's just understand, or let's just remove this methane molecule from it. Let's just write ligand, okay, so that it will be very simple for us to understand and no confusion will happen. Let's just write ligand molecule, okay. So we can assume that this ligand molecule will have either of these two things, okay. Either it will have a HBA, that is hydrogen bond acceptors, and or either it will have a hydrogen bond donor, okay. So the ligand will possess what you can say some groups which will be acting as hydrogen bond acceptors, or they can act as hydrogen bond donors, okay. So these two things that we see here, okay, these play an important role in doing the interactions. Now, since the ligand has it, don't you think the residues should also have this? So let's just write here amino acid 1, amino acid 2, amino acid 3, amino acid 4. So these amino acid residues will also be having something which is called as HBA and HBDs with them. So we can also write amino acids having HBA and HBDs. Now, as soon as these HBAs and HBDs, see, we all know through basic chemistry that a hydrogen bond except our group interacts with a hydrogen bond donor group. So if my ligand has a hydrogen bond except our group, so it is bound to interact with a residue which is having hydrogen bond donor group, right? So by this, it will form an interaction. Okay, let's just say I will represent this interaction in the form of a dotted lines. Okay, it will form an interaction. It might also form an interaction with amino acid 3. Okay, like this. So like this, it is forming some interactions and sitting inside the pocket of the protein. Now, it is not at all compulsory that only hydrogen bonds exist in the pocket. There can be other interactions as well. It can be van der Waals interactions, okay? You might not see a proper interaction, but you might see a van der Waals interaction. There are many interactions that we have studied, right? So you might have that as well. You might also experience some salt bridges that are getting formed. We'll not go to the depths of it, but we should know that how does a ligand sit inside the pocket, okay, and forms an interaction with the ligand. So I think this is clear. So we know, for now, we know that ligand comes inside the pocket, sits there and forms an interaction. So what are we, like, this is done. PPT is not visible. Sir, I am explaining on the board. So yeah, yes. So now you can see, you must be thinking that this is all like about docking, right? So where does the simulation comes in the picture? So all this work, taking the ligand structure, docking inside the protein pocket, studying the interactions, which residue is interacting with which group of the ligand. This is all docking part. So where does simulation come into the role now? So here, if you clearly see, we are keeping the protein as a rigid structure. Ligand will be taking different conformations when we are docking, right? You must have heard about some softwares. Some enthusiastic people with us who have joined might have already studied some softwares like MOE. Then we have Autodoc. There are various softwares. What these softwares do, they apply various algorithms, and then what they do is they apply these set of rules to the ligands and the protein. The protein will be basically rigid. Okay, it won't move. The residues inside it won't move, right? But the ligand will move. The ligand will. It's like if this is my hand, right? If this is my hand and I have curled my hand in this way, I formed a pocket inside this, right? So for this pen to fit inside this, now this pen is not able to fit inside it like this, right? So it needs to mold itself in such a manner and it fits inside the pocket. So what my docking software is doing is it is trying to take up different conformations of the ligand and trying to fit this ligand as a key to this lock, right? So it will take this and insert this in the form of a lock, right? It's a lock and key interaction that we see here. So this is docking. And in this, if you see, there's a limitation. The protein isn't moving. But what we know is inside our body, everything is dynamic. Nothing is rigid, right? Everything in our body, if you see, if you consider a cell of our body, it is basically consisting of cytoplasm. What is cytoplasm made up of? It is a liquid stuff, right? It's liquid. And in that, our organelles, our various components of the cell will be floating inside it. Then the cell will also be having some proteins inside it. The cell will be also having some membranes. Like if you know that basic biology that we know is that the cell will be having a cell membrane around it. So on those cell membranes, there will be some receptors that will be uh, situated there. So if I could just represent it here, if this is a cell, so this is the cell membrane. And here you will be having some receptors through which the ligand will interact and cause some secondary interactions. Okay, so there will be a cascade of some chemical stuff that will be happening inside the cytoplasm and then leading to some biological activity. Okay, so we'll not go into that. we have to just focus on this receptor here, on this protein structure here. So if you see, this protein structure is embedded in the membrane. Okay. So if you can see this, it is embedded in the membrane, but it is constantly moving. It is moving by the sense it is constantly having different conformations. Okay. If you're more curious about this, you must have seen our flyer for today, right? In the flyer, I'd shown some simulations. If you would like to see that, I will show that at the end of the simulation part. You could see that proteins moving inside, right? So proteins are dynamic in our body, and they're trying to adopt different conformations. They're not going to be the same because they're floating, right? They're floating inside the cytoplasm, or maybe they're floating inside the plasma of our body, right? So if you think deeply, if the conformation of the protein, if the structure of the protein is changing drastically, what will happen that way that the ligand interactions will change drastically? Okay. Suppose if this my hand is doing such movements like this, okay, and I've put a pen inside it, won't you think that the pen, okay, which is trying to interact with this pocket inside, will have different interactions at different points? Don't you think that? So just try to feel it. Because if I try to fit this pen inside my hand, inside this pocket is moving constantly. The area of the pocket is constantly changing. Don't you think the interactions of the ligand with the residues inside the pocket will also change with time, right? So in simulations, we're trying to uh, prove, or we are trying to negate this limitation of docking software. In docking, we have kept everything rigid, but in simulations, what are we going to do is we are going to keep everything dynamic. The ligand is going to move, the protein is going to move, and through this entire simulation pathway, what we're going to show, okay, so we have done a simulation of 1 nanoseconds. I'll come to that. What is 1 nanosecond? What is 2 nanoseconds? What is 3 nanoseconds? Okay. So we've done a simulation of 1 nanosecond, 2 nanoseconds. And then from there, we will analyze that throughout this 2 nanosecond journey, or throughout this 1 nanosecond journey, how did the ligand change its interactions? Or we can say, how does a ligand try to interact with the changing pocket? Okay, with the changing, what you can say, the changing area inside the pocket, right? So how does a ligand do that? Because inside the body, everything is moving, right? So it has to adopt itself accordingly. This is one part of the simulation that we'll be covering. The second part is, inside our body, we know that there's a particular temperature that is maintained. There is a particular pressure that is maintained. So how do we mimic that? In docking software, it is not mimicked. So in simulation, we are going to do that. In simulation, when we are basically doing simulations, we have to set a particular pressure. We have to set a particular temperature. So according to your body, what's the pressure? It is 1 bar, right? It is 1 atm. So we need to set the simulation box in such a way that the pressure is 1 atm. We need to set the temperature in such a way that the temperature is 35 degrees Celsius or 37 degrees Celsius. That is the room temperature, right? Or the body temperature, what you can say. So we need to set these conditions. And what are we doing is we are going to mimic what is existing inside our body. So if I could just show you in the form of a diagram, you could see this. So what are we going to do this? We're just going to visualize how does a simulation box look like. Okay, so everything we are doing in a computer, it is going to be virtual, right? So virtually we are going to form a box. Okay, inside that box, we are going to place our protein, and inside that protein, we are going to place our ligand. Okay, so that is just see this how I'm doing this. Just see this. It's a 3D box. So for now, I'm drawing a 2D box. Okay, let's just imagine that this is a 2D box. Okay, inside this 2D box, we're going to place our protein structure. Okay, and inside it, we are going to place a ligand. Let's say this is a ligand. Okay. So this is a ligand that is fit inside the box. So we're going to take this box. It's a protein structure here. There's a ligand structure inside the pocket. And we're going to place water molecules all around. You can place water molecules. You can place other solvents also. But inside a body, what is it? It's water, right? Body is 90% water, 70% water. What do you say? So we're going to fill this box with water. Okay. So we'll have some water molecules here. HOH, HOH, HOH. Okay. So this box will be filled with HOH molecules, water molecules. Because what we know, our protein is not existing alone. It will be having several chemicals all around it inside our body. Our body is full of chemicals, right? So this protein structure that is moving inside our body, it will be surrounded by water molecules. It will be surrounded by many other molecules, many other chemical messengers that we can see, right? Or our ligand molecule for that matter. Okay. So the protein, along with making interactions with the ligand, will also try to interact with the water molecules. And this is where it covers the fourth limitation of docking surface, because in docking surface, you're not able to insert water molecules, right? Okay. So how do we study that in when our protein is existing with water molecules? How does a protein then do interactions? How does the interaction of the protein change then? So because water molecules can also interfere, right? You will also learn in the future that some protein molecules need some water molecules all around. Okay, so this protein structure, this table will require 2, 3 water molecules around for it to be stable. Okay, let's just say that this is my protein structure. This is my secondary protein structure. Okay, or tertiary, whatever you say. The structure, for the structure to remain stable, there will be some water molecules inside the pocket. There will be some water molecules outside the pocket or surrounding the protein. And what will they do is they try to interact with the protein. If I could just draw it, you will understand it better. Okay. So if I could just draw it, suppose this is my protein structure. Okay. And I have HOH, then I have HOH. Okay. Water molecules, HOH. Okay. So I have these water molecules around this part. So if I can say that this is amino acid 1, this is amino acid 2. So what is happening is this second structure that you see here is interacting with this water molecule. This is interacting with this water molecule. There's an HOH molecule inside the pocket also. So there will be an amino acid. This will be interacting with the water molecule. So these interactions that we see also have to be studied. And this can, what you can say, change how the protein looks like. Okay, because this sometimes what happens is the protein structure without these water molecules cannot really maintain its structure. So for it to maintain its format or for it to maintain its coiled structure, it will require some water molecules to form a stable interaction, right? So water molecules are very, very important, and that is covered in simulations. Okay,